Good evening, everyone. So, this week we'll be discussing vaccines in HIV, and this shall be the last lecture in the HIV series till now. We might visit again HIV after many months or some time has passed. And this, I think, is the 10th video in the HIV series, and we have covered very important topics in understanding the concept of the immunodeficiency disorder that is caused by HIV. Now, this would be a bit dry lecture because all you have to do is just memorize one or two lines of few words to know what types of vaccines were tested. But I have seen many questions coming in the exams on short notes on vaccines and HIV. So my job this week would be just to keep you apprised about what all you can write about and what all should you mention if that sort of a question comes up. And this kind of completes our discussion. We have discussed in the previous weeks regarding the structure of the agent, then how the agent behaves in the body, how does it cause immunosuppression, then the treatment and all the major classes of drugs along with newer drugs that are in the market. And now we'll just discuss vaccines in HIV and their impact on humans and in animal models. And after that, we will finish and move on to the next series of lectures tackling different topics apart from HIV. So without much further ado, let's start our discussion this week on vaccines in HIV. Now, as of now, there is no vaccine for HIV. Then that is an issue because we have, in fact, we have multiple vaccine candidates, but we don't have a proper working efficacious vaccine against HIV. And that is why prevention of transmission is the cure. To prevent transmission from one individual to another individual is what we have to restrict as of now because we don't have any effective vaccines that can prevent the onset of infection after the agent has entered your body. There's no protective immunity as such. Okay, so the major onus of prevention lies in the prevention of transmission from one individual to other, and that is why a lot of stress is being given regarding your checking of blood samples while donation or all the contraceptive methods during sexual intercourse. For a vaccine to be successful, it has to be safe, it has to be effective, durable, and protective immunity has to be established, and there should be a memory also to prevent infection and or disease. Now we'll understand these words very quickly. It has to be safe, tolerated in human population. It has to be effective. That means proper immune response should be there. It should be durable. We know that the HIV infection, once it has set in, will remain there for life. So it has to persist throughout life to provide a proper immunity against the virus. The immune response that is elicited from the vaccine should be protective enough against the HIV and to prevent any infections in a non-infected individual. So if an individual, okay, if you have an individual and the agent has entered the individual, let's see, the agent has entered the individual, a vaccine should prevent the establishment of infection. That is, it should prevent the integration of HIV genome inside the host genome. Plus, if the individual is not been infected by the HIV virus, a vaccine should be able to prevent the virus from entering the cells or destroying the cells. Clear? Let's move forward. There are many obstacles in making a good HIV vaccine, and these are the kind of obstacles that we should know about. First of all, the type of immunity is unclear. We know that HIV infects, the prime target is CD4 positive cells, T lymphocytes. But the issue is what arm of immunity can help to provide a good enough protection against HIV? Is it the innate immunity, adaptive immunity, natural killer cells, antibodies, or macrophages? What arm of immunity can provide a protective shield against the HIV infections? That is a bit unclear. The HIV proteins are highly variable. They are very mutating. They mutate, change their structure, change their protein configuration, and because of that, if we take an antigen from HIV virus and make a vaccine against it to protect the individual, the protein mutates, and the HIV virus is able to get surpass any kind of protective immunity. Multiple vaccine candidates that have been tried have found to show no efficacy in trials, and these are mostly in the human volunteers. Okay, human volunteers. While multiple vaccine candidates have shown efficacy in animal models, mainly monkey models and macaque models, but in human volunteers, they're not shown to be very effective. There are logistic issues. how to make vaccines. Vaccine making is not a cheap task, it's very expensive. So how to make vaccines, if a vaccine candidate is being found to be efficacious, how to supply it to everyone else, how to supply it to all the at-risk population, all the individuals on the all the humans in the world. Vaccine research, like any other research, requires funding, and even HIV is such a implicating disease, or such an important disease of international dominance. The funding is not that much so as to have proper care in developing newer and newer vaccine candidates. The cost is also an issue. That means when the vaccine is formed, would it be easily accessible to patients, or will be sorry to individuals? Will be easily be able to People will be able to buy it easily? So that is also an issue. Acceptance, for example, if a person is vaccinated, do they accept that now they have immunity or not, or do they accept that there is any protection against the virus, or it is regarded as a useless immunity? So that is one thing. And the other issue is that increase in high risk behavior. Let's say we have an individual and there is a very efficacious vaccine available against HIV, and we vaccinate that individual. Would it increase the thought process or the psychology in that individual that now I can indulge in multiple high risk behaviors because I'm vaccinated, nothing will happen to me? So, vaccination can increase high risk behavior, and that is a major issue while developing HIV vaccines. So, stress is also made on limiting or curbing this high risk behavior, and that is why as of now the prevention of transmission is the only cure. So this point becomes much more important in the absence of an effective HIV vaccine. Clear? Let's move forward. Good evening, everyone. I hope you're enjoying this video, and I've come again to ask you to vote for Delhi for Dermacon 2028. So, with the support of IDB Delhi State Branch, do vote for Delhi and come and meet all the dignitaries here, and let's have a good academic discussion in the backdrop of the cultural heritage that Delhi offers. So, enjoy this vibrant city that I duly call my own, and if you follow this instruction to become an e-voter, you can just easily vote on the IDBIL website. It hardly takes less than a minute, and it's better to vote as early as possible. Will help you in tackling all those spam calls that you get. So make sure that you choose Delhi as a good venue in 2028, and let's meet there. I shall see you in Delhi. Bye bye. Now, let's start our discussion on vaccines. There are multiple vaccines available and tested for HIV. In fact, and there are so many trials going on with thousands and thousands of patients currently ongoing, disbanded in between because of lack of efficacy, completed their study and found the vaccine to be not much effective. Newer candidates coming. So we have so what I've done is I've divided into separate parts so that you are able to understand what all topics have to be discussed. So if any short note comes on vaccines in HIV, make sure you go through all of these 6 headings so you are able to add it. Okay. A seventh heading would be mRNA based vaccine, but these are the latest vaccine that is developing. A lot of research is going on this year. Multiple candidates have come. So if you want, you can add that into the list. The vaccine includes live attenuated vaccines. That means live HIV virus, but you have decreased the efficacy, decreased the infectivity, and used it as a virus. Inactivated vaccines, that means you're using inactive HIV virus. Virus-like particles, that means you're using particles, parts of viruses. Subunit vaccine, you're using entire unit of vaccine. For example, you can use GP 120, 41, or 160 units. Okay, you can use P24. So all of these units have been made. DNA or genetic vaccines, in which you have a bacterial vector, and inside the bacterial vector you induce HIV genome or gene or the entire RNA, and then the bacteria translates it, forms protein, and express the proteins, and these proteins are recognized by the immune cells or the T cells, immune cells. So these are DNA genetic virus, and then live recombinant or vector vaccines, in which the bacteria has genes inside which encodes for HIV antibodies. So let me explain that. In the DNA and genetic vaccines, you have a bacterial vector, and inside you have the bacterial genetic material, and inside the bacterial genetic material you have the HIV material. When this gets translated, it will form HIV antigens. which when expressed on the surface will be recognized by an immune cell and the immunity starts. That's how DNA-based vaccines work. In live recombinant vector vaccine, instead of having an HIV genome in between, what you have right now is a gene that encodes for anti-HIV antibodies. And these antibodies will be made by the bacilli, okay, by the bacteria and secreted. And these antibodies will help in fighting the infection against HIV. So these are recombinant vaccines, so live recombinant vector vaccines. Clear? Let's move forward. So let's discuss each of these vaccines in detail. You do not have to remember much of it. You just have to remember the 6 headings, 7 if you count mRNA. And if you just remember the headings, if you want, you can remember 1 or 2 candidates from it. But I think the vaccines heading is enough. And you can write few lines about it and easily write your answer on vaccines in HIV. The first is live attenuated. Now this vaccine has been used in animal models and is found to be effective in animal models but not in human models. In this you have the NEF deleted attenuated SIV. SIV is simian immunodeficiency virus, which is closely related to HIV, and in the SIV you have deleted the NEF gene. NEF is a very important protein in HIV, and if you want to know more about NEF, you can go and see the structure of HIV video in which we discuss NEF. So NEF, you have to delete the NEF and you have attenuated the SIV and now you're using it as a vaccine. Another candidate is a non-pathogenic SHIV. Now SHIV is simian and human immunodeficiency virus, which is a a combi virus, a dual virus which infects both. Second is inactivated vaccine. So we have discussed the first one, live attenuated. The second is inactivated vaccine, in which you inactivate the virus HIV by HIV and then inject it. In inactivated vaccines, you remove zinc ion from GAG and INT proteins. INT is integrase proteins. Okay, so in GAG and integrase proteins, you remove zinc ions, which is required for proper formation of HIV virions, for proper activity of integrase enzyme. We have discussed a little bit about in the structure of HIV video. Another way to inactivate virus is to use sorelin and UV inactivation of viral RNA. So if you know about sorelin, so what sorelin do is under the action of sunlight or UV light, it creates DNA and RNA adducts. And that is how we use sorelins as an immunomodulator or immunosuppressor in vitiligo and psoriasis. The same thing we can do with HIV and inactivate the virus and make a vaccine out of it. Clear? That's the second vaccine, inactivated vaccines. Hi. Hope you're enjoying this video. I know it's a long video, so I thought I'll give a certain break after listening to such a long videos that I make. If you think that these videos are helpful in your residency, do consider becoming a member. You can opt for Sapphire membership or Ruby membership. I would prefer it to be a Sapphire membership, of course. You get access to longer videos, bonus contents, extra information to make things more interesting, and to cover each and every aspect of the topic that we are discussing. Okay, so I think that's enough of a break. I should continue with the video, and you should listen to the other person at the bottom right corner tell you different topics of dermatology, venereology, and nephrology. That being further said, let's continue with the video. The third is virus-like particles. Virus-like particles contain only the some parts of the virus. They only contain viral core or envelope proteins in another virus vectors. So what you do is you have an HIV agent, you have a protein, you take a part of protein, put that in another virus, and use this as a vaccine. This is virus-like particles. These vaccines require DNA vaccine priming. I'm not going to detail what is DNA vaccine priming. Essentially, you have to boost the immune response by giving another vaccine along with it. So virus-like particles are mostly we use envelope proteins, and we have found in multiple animal studies that using envelope proteins lead to a broad-based cell-mediated response. So a broad neutralizing antibodies are found, and that is why they are better. A concept is chimeric virus-like particles. That means you have HIV proteins coupled with other virus surface antigens. So you are using two antigens from two viruses. One is HIV, one is another virus. And the another virus that you use is a virus that we already have a vaccine against. Okay, so you have the HIV protein, you couple that with another virus protein. This we have a vaccine for, and this is HIV. And then you inject it as a vaccine. The immune system, since it already recognizes this vaccine or capable of recognizing the vaccine, also starts to recognizing HIV. So an example of that is to combine HIV surface antigen or HIV proteins with hepatitis B surface antigen. So this is chimeric vaccine-like proteins. Let's move forward. Fourth is this subunit vaccine, and it is one of the most important vaccine candidates. We have a lot of vaccine candidates from this particular heading. The first is native envelope proteins, in which we use envelope proteins like GP160, 120, or 140. We have to utilize an adjuvant. Adjuvant is a non-biological substance which is added to the vaccine to induce this immunogenicity. So adjuvant we use alum, lipopolysaccharide, deoxycholate. These are all adjuvants that are used in HIV viruses. The neutralizing antibodies that are formed is against hypervariable V3 dominant GP120. In fact, GP120 is one of the major vaccine candidates in HIV, but it is also the major mutable protein, a protein that undergoes a lot of mutation. So that is one of the difficulties in making a vaccine. We can create monoclonal antibody against the V3 loop. So if you have gone through the structure of HIV virus, we have discussed that if this is GP120, this is GP41, there is an area here V3 loop of protein, which is helpful in adhesion of HIV virus to the lymphocytes and also in adhesion to the CCR5 receptor and the CD4 receptor. And when that adhesion happens, GP120 goes away and GP41 becomes a hairpin loop and the virus enters the cell. All this have been discussed in way detail in previous videos. Go through that. Let's move forward. Coming to the next heading, we have DNA and genetic vaccines. We have discussed that in the introduction video. The bacterial plasmids, we use bacterial plasmids, or we have bacteria and inside we have the circular plasmid, which is just a circular genetic material. And here in DNA and genetic vaccines, these plasmids are used to induce HIV antigens for immunity. How it does that, we have discussed very shortly a few slides back. In the sixth type of vaccine, we have the live recombinant or vectored vaccine, in which we use live viral or bacterial vectors that carry gene for desirable HIV and SIV antigens. Okay, so what we are doing exactly like that. In DNA and genetic vaccines, we use the genetic material to have the antigens. While in the live recombinant, we have the whole organism, the whole bacteria encoding genetic material which is required to make antibodies against the vaccines, antibodies against the HIV. Mostly, the vectors that has been used are the avian pox viruses. So viruses like canary pox, fowl pox has been used. Other viruses like vaccinia virus, the modified vaccinia ankara, don't remember these names, just remember that avian pox viruses vectors have been used. Adenovirus and bacilli like salmonella, the complete salmonella has been also used as a vector. Let's move forward. So without much further ado, we have finished the good talk about vaccines in HIV. These are some reading recommendation. In fact, after this video, you don't require, this is enough at residency level so that you are able to answer questions in Viva or write a short note. Okay. In fact, I would like to recommend this article. This is a very good article. Okay, gives a proper detail about how to develop not what are the features of a good vaccine should be, and what are the vaccine candidates that have been tried. Some have failed, some have found to be efficacious. What are the immune correlates of infections? How do we check whether they're efficacious? It's a very good article. If you want to read casually or want to have more knowledge, you can go ahead and search for this article. So again, I would request you to become a member of the channel. That helps me to motivate and make videos week after week for years altogether now, not 2.5 years or something. But do remember that I do plan to discontinue Ruby membership in the coming months. And if you really like these videos, I would request you to become a Sapphire member of the channel. The Sapphire member have unique earlier access to videos, and in fact, multiple times I put polls to decide what all new series that we should begin after completing one series. In fact, a poll is going on for right now for Sapphire members, in which I've asked them what topic should we tackle next, and the connective tissue disorders is winning the poll as of now by a huge margin. About not two-third, about three-fourths of people have voted for CTDs. So maybe we'll discuss that next time. Okay, so without much further ado, I would like to stop, and I will try to stop before we reach 40 minutes for the members. For the general public, you will have access to shorter videos. So adios and bye bye. I shall see you next week with a newer topic.